Okay, so today you read all about energy and energy transformations and the way in which we harvest the energy from our food and bring it into different usable forms in our body. So lots of great questions came up. And one like really fundamental question that I had to take a minute and think about was somebody asked, but why is matter not destroyed? Which was sort of like a deep philosophical question, if you ask me. And my answer to them was that you can't take something and make it into nothing. Something is always going to remain of what was behind. So even though sometimes from the outside, chemical reactions might look like something's disappearing, like when we burn a candle, it looks like that candle has disappeared, but it hasn't. It's just been rearranged, and it's been rearranged into molecules that look different than what we saw before. And sometimes those are molecules that float into the air and they're invisible. So anytime there's an interaction happening between molecules, there's rearrangement, bond gets broken, bonds get formed, but the elements, the atoms that were there in the beginning are always still there in the end. You read a lot about different kinds of energy, and these are the two kinds of energy that we see the most, at least in biology. So kinetic energy, which is energy of movement or energy of heat, and then potential energy is energy that's stored in location. So the example that was given is always given is something at the top of a hill. Something at the top of a hill has position that it can roll down. In biology, the position that we talk about is electrons, and it's their position relative to one another. So I am not going to explain all of what that means, but I will briefly tell you that, we talked about last week that there are covalent bonds and electrons are shared, and that sometimes those bonds are spending more time around one atom than the other. So when that happens, the position of those electrons gives that bond more or less energy. So if electrons are held in the very center of something, that has more potential energy than something that's held really close. So you also read about all the different ways that energy is transformed in these different categories and terms that we apply to them. So I brought this as like a summary chart to try to put it all together in these two categories, because it gets confusing with all the different terminology. So we essentially have pathways and reactions that release energy and those that require energy. So on the top, these are the ones that release energy. So they're exergonic. Exer means out. So they're putting energy out into the environment. We call these catabolic. Catabolic, I always remember that cats destroy things. So catabolic reactions often break stuff down, right? So a lot of the reactions that we think about with digestion, when you eat a big hamburger and you break it down into little amino acids and sugars, right? Those are all catabolic reactions. They often require water. And you read about how when you break down those big molecules, the water goes in and splits them, right? So all of those hydrolysis reactions, they're all in this category. And then this is the word that threw a few people on this reading, which is that these pathways are also spontaneous. So spontaneous has lots of different meanings. I actually looked it up in Webster yesterday. And several of the meanings like relate to what is actually meant by this. And so what this means is that spontaneous can be something that occurs without an input of external energy or force. And so we're not putting energy into this reaction. It can occur without any force driving it forward. So that's what it means by being spontaneous. So this graph is showing, trying to show you this difference in energy. So reactants in this case have a lot of energy. So those bonds, whatever bonds they have high potential energy. And the products of their reaction have a lot lower potential energy. So this energy difference, this is how much is released. And it can be released in different ways. The opposite of that are anabolic pathways. So everything that I just said, except in reverse. They are endergonic. Ender means in, so you're putting energy in, so they require energy. They're often building something big from something small. So they're going to take those amino acids and build them up into a big protein, and that requires a lot of energy, right? They often release water. And then this graph is just the opposite. The reactants have lower potential energy. When we stick them together, the product that comes out has higher potential energy. So this amount of energy had to be put into the system to make that go forward. So how does this all happen? So the truth is that even things that are spontaneous reactions won't happen on their own on a time scale that's appropriate for life. So in our body, all of these reactions that are happening, if there were no enzymes, eventually these things would happen, but it wouldn't be at a rate that would be sustainable for you to maintain a cell. And so the role of enzymes is to speed up all of those reactions. It doesn't matter whether we're talking about a reaction that's going to release energy or require energy. What an enzyme does is it sort of gets over this initial hump that's required to bring things together, and we call that activation energy. So this graph is trying to show you the same thing that this is, except it's adding activation energy. So in this case, reactants are high, products are low. Reactants are higher, products are lower. But even though this reaction is going to put energy out, there has to be a certain amount of energy put in. And what that looks like, this is all very abstract, and that's how it's often written. So I brought this to show you what that really means. So the energy that has to be put in this abstract example is the bringing together of reactants. And so what enzymes do, they have these spots where the reactants are going to bind. And when that enzyme changes shape, it's going to put force on bonds. It's going to push things together. It's going to bring them in the right orientation to overcome this barrier that they might have to create this bond. So one thing that's important to remember is that anytime atoms are coming together, it's the electrons that are on the outside, right? Electrons are negative. So anytime two atoms come together, there's a moment where they're repulsive because you have two negative charges bouncing off each other. And so the role of an enzyme is often to overcome that and shove them together anyway. So that's delta G. And action where you're releasing energy, this is going to be negative because it's going down. And when we have to require energy, it's going to be positive because it's going up. And if we were in a chemistry class, we could calculate all of these things about entropy and delta G and the actual energy of these different bonds. Somebody actually asked, like, is there difference in the energy in bonds in the book? And I talked about this idea of like where the electrons are positioned. So whether something's polar or nonpolar impacts its energy. Nonpolar bonds have way more energy because of that position. And that's also why fats have more calories than proteins, because their fats are made up of nonpolar bonds. There also are differences, going back to this one, whether or not it's a single bond or a double bond. So that double bond stores more energy because there are more electrons involved in that reaction than a single bond. So all kinds of different things factor into how much energy are in these reactants and products. And delta G just describes like how they're going to, whether it's going to be increased or decreased. Let's talk about enzymes a little bit more. So we just said that their role is really to just get over this energy hump. And you read and had lots of questions about enzyme inhibition. So this is showing you three different ways that an enzyme might be interacting with its substrate. So substrate is just a reactant. So ordinarily, this reactant is going to fit nice and neatly into an active site because the molecular arrangement of that substrate and the enzyme are so that they fit just together, right? And when that happens, the enzyme will change shape. Just like we saw here, it'll change shape, it'll make something happen, and it'll release products, right? So in inhibition, there are a few different ways, and your book talks about these two, the idea of competitive inhibition and non-competitive or allosteric inhibition. So in competitive inhibition, that means that a molecule is binding in the active site, and now it's in the way. So one of you said, hey, is this the same thing as caffeine? Yes, it is. It's the same thing that we talked about a few weeks ago. So here's our adenosine receptor. It's also got enzymatic activity. We call it a receptor because it sits on the outside of a protein or outside of the cell and binds stuff. So it is receiving stuff, but it's also got enzymatic activity. So here's your receptor. It's got an active site. Normally, adenosine can bind. You feel drowsy over the day. But if you have caffeine, then caffeine is binding instead. The adenosine can't bind, and so you're not as drowsy. So that's exactly what's happening there. The other side of this is non-competitive inhibition. So what happens there is that something binds the enzyme in a different spot. So it's not that it's taking up this slot, it's that it bound somewhere else and fundamentally changed this spot. So now this looks different and this no longer fits. So someone asked for an example of that. And the example is aspirin. So this is how aspirin works. There is a receptor, another enzyme called COX-2, and it releases these molecules called prostaglandins. Prostaglandins are a major source of pain. So prostaglandins are, I know this because I teach sex and reproduction, the main reason why people have menstrual cramps, prostaglandins, you can blame them. But they're also the reason for like lots of other pain in your body, including headache pain. And so if you can block the production of prostaglandins, you can have less pain. So this is our COX-2 enzyme. And normally there's this molecule that gets released when something's going on in your body that's going to trigger pain. It's called arachidonic acid. So here's your AA. And normally it can bind to COX-2, and then COX-2 releases prostaglandins and you hurt. But when you take aspirin, aspirin binds to COX-2 in a way that means that this can no longer bind. And so you have less of this pain molecule being released and you feel less pain. So lots and lots of drugs that we take are acting in one of these kinds of ways, because a lot of the stuff that's happening in your body when you're in a disease state or in pain is because of some misregulation or overactivity or underactivity of those enzymes. And we can affect those, by adding these different inhibitors. All right, I'm pausing for questions. I just told you all about enzymes and energy. And in a moment, I will talk more about ATP and cell respiration, but I want to pause here. So you have to have proteins to make enzymes. All of the things in your body that are enzymes, they're proteins. And so you have to have protein intake. So one of the things we talked about last week was that if you are deficient in protein, your body's going to get it somewhere. And if you're deficient in energy overall, your body will pull from carbohydrate stores in the liver, right? And we store fat in our adipose tissue. We don't store protein, but your muscles are a big source of protein. And so this is why if someone doesn't consume enough protein, then they'll start to have muscle wasting, because the body is going to get those amino acids from somewhere, because it can't function without these enzymes. Yep, so that's an important reason why it's absolutely necessary to consume protein. Also, I'm vegetarian. I've been a vegetarian for 26 years now. 26 years. I ate tofu this morning, I swear to God. It's delicious. But there are also lots of protein in other things besides just meat, eggs, and tofu. Because if you think about the fact that we just said everything that's doing all these chemical reactions are proteins, right? Every cell in the world is full of proteins. So that apple is full of proteins. It's not nearly as dense as like a steak, which is all protein, but there's still protein even from food sources that you wouldn't think of. And things like beans and pulses and legumes have an even higher concentration of protein in the parts that we eat. So there are lots of food products that you can get good protein from. So this is trying to show you illustrate in a couple of different directions, the idea that the concentration of the stuff that's binding to the enzyme, so in this case, starch or whatever the substrate is going to impact how that enzyme behaves. So if there's more starch than there is enzyme, then the enzyme is going to be maxed out. It's operating at its maximum capacity. There's way more starch around than there are enzymes to actually do that work. Versus when the substrate concentration is low, then adding more enzyme increases the rate. So in the beginning, that's when there's maximum starch, right? Nothing has yet been broken down. And so there's so much starch around that all of the enzymes are saturated. Every single enzyme is working. And so the rate is really fast. But as that starts to break down, the starch concentration also goes down. And so that means that there are some enzymes that are sitting around without any substrate bound, just because the concentration is less. So the overall rate is going to go down because some of those enzymes don't have anybody to digest. So when you eat food and your stomach and small intestine are receiving that food, there are all kinds of chemical signals and hormones that are being released to tell the pancreas and the liver what kinds of enzymes we're going to need here. And so those enzyme levels will go up because there's a bunch of food in the system. And that means, yeah, there's a bunch of substrate. We need a bunch of enzyme here to break this stuff down. And then when there's not a lot of substrate around, then it's a waste of energy to continue making so much enzyme. So the enzyme levels will decline. So the later parts of this chapter were all about cell respiration and how your mitochondria are playing this role to create ATP from the energy that's stored in food. So here's ATP. ATP has this piece over here, which is adenosine, which we also just talked about. This, if you were to take one of this off and link these together, this A is also part of RNA and DNA. But when we have it in this form with 3 phosphate groups linked together, this is our energy source. So this is the molecule that's the primary energy source for all cells. And the reason why it stores so much energy is because of these 3 phosphates. So each of these are negatively charged. You can see there's little negative ions here. And so just like anytime 2 negative things come together, they're going to repel, right? this isn't just the one electron. This is this whole big giant thing with the phosphate and four oxygens around it and all the electrons that come with it. And so it's highly negative and big. And so putting two of these together is hard, and it requires a lot of work to get them together. So when you form that bond stores a lot of energy. And it's extra hard to get the third one on after you've already got two stuck together. So that bond stores even more energy. So releasing this bond by taking one of those off then is a form of releasing energy, and that energy can be used for other kinds of reactions that need energy. So just like up here where we said some reactions are going to release energy and others are going to require it, ATP, the hydrolysis of ATP is happening all the time. And so it's doing this, it's releasing energy, and that energy is then harvested by all the other reactions in your body that need it to be put in. All right, I sort of just said all of this, and this is just showing you that we're going back and forth between ATP and then ADP. So ADP is this when we have two, so this is di instead of tri, and this has lower energy. So when we take a phosphate off, we're releasing energy. When we put a phosphate back on, we're putting energy in. The other player that you read a lot about is NAD and NADH, and this is an electron carrier. So its job, it's like a taxi. Its job is to shuttle electrons from one place to another. So when it's empty, it can pick up electrons from glucose or other molecules that are being broken down in your body. So as that glucose molecule, which stores lots of energy, is broken apart, it's releasing electrons, and they are holding that energy that's been released. So those electrons then get picked up by molecules like this, and then they can shuttle it off to another place and drop it off at a place where it's needed. So these are our two big energy exchange players, and they are involved in all of the steps of cell respiration. So I brought you a couple of different ways to look at this because I know that this is like a really complex idea and there's lots of steps. So first, I do not expect you to memorize all the steps or how many of everything is in. Also, your exam's open book, so like you can look this up. And what I'm more concerned about is that you sort of understand the concept of what's happening and where the energy is going. So here's one example, and this is showing you the mitochondria, and it's trying to show you like what's happening where inside of this awesome organelle. And here are our 4 steps. So we've got glycolysis. This is the first step. It's happening outside of the mitochondria. So even organisms that don't have mitochondria, like bacteria, can still do this because they don't need a mitochondrion to do this. All this other stuff in here is happening inside of the mitochondria. We have this first step that is often glossed over, which is just like a transition step where we take this product and put it in here. Then this is the citric acid cycle, also known as the TCA cycle, also known as the Krebs cycle. They are all the exact same thing. That happens inside of the mitochondria, and its main job is to make a whole bunch of these. So these are our electron carriers, a whole bunch of them get made here. And then this last step is taking all these electron carriers that were made before and then shuttling down this chain to make a whole bunch of ATP. So just looking at this, looking at like these 3 big stages, this first stage, its job is to break down glucose. You got glucose, let's do some initial breakdown and see how we can use this. So that's what's happening in glycolysis. You get a little bit of ATP, you get a little bit of energy in the carriers. This is to take this energy or to make lots and lots of energy in the carriers. And it also completes the oxidation of glucose. So glucose, as it breaks down, gets turned into CO2. So by the time it's all passed through here, it's all gone. All that glucose is gone. It's now in the form of CO2. So at the end of that, you've got all of these, you've got a couple of ATP, and then the role of the electron transport chain is to harvest all of this energy that's in those shuttles, those taxi cabs, and bring it to a place where it can make a whole bunch more. If you like your information more stepwise and not so artsy, here's the same information. Here's step 1, we're getting a little bit of ATP, some NADH, we're breaking down glucose. This is the step, this is what it gets created into, pyruvate. So pyruvate then gets converted to this, acetyl-CoA. We make a little bit of NADH, we pop one of the carbons off, and then this cycle completes breaking down the glucose, makes more of these, makes a little bit of ATP, and it's all brought over here. So I want to ask you a question. Given what I just said, and looking at our equation overall. So this is the overall sum of the equation of what's happening when we go through this process. So this is glucose, we combine it with oxygen, and we get out carbon dioxide and water. So these are where the atoms are moving around. So at the end of this first stage, before we get to the electron transport chain, where are all the atoms from glucose? And where is all the energy from glucose? So first, let's talk about atoms. Where do you think the atoms are? So where is this carbon? Where does it end up? It's ending up in CO2. So through both this intermediate step and through the citric acid cycle, we're moving these carbons from one place to another. So the very first thing we said was nothing gets destroyed, right? The carbon has to go somewhere. So this carbon ends up in CO2. So then where's the energy? Because we said that the energy is stored in the bonds, right? So when we break these bonds apart, there was energy in those bonds, but we also have to account for all of that energy. So where's the energy now? Most of the energy is in here. There's some energy that has been produced and has been put into ATP. So there's a little bit of ATP here. There's a little bit of ATP here. CO2 has some amount of energy in it. So there's still energy in the bonds between the C and the O, but it's much lower than what was in glucose. Then most of the energy is in these carriers. And it's these carriers that then are the thing that is the input for this last step of the electron transport chain. So before we get there, we've taken care of all the atoms, they're here. And all of the energy is in this form where we're about to put it in to the electron transport chain. All right, this is just another summary. I told you at the beginning, I brought you many slides that show things in different ways. And this is just summarizing, this is what we sort of got out. We got out some electron carriers in ATP, we got out electron carriers in CO2. Here we got all three. And now all of these things are going to be put in to the electron transport chain. Okay, so we said that all that energy is sitting in electron carriers, and that electron carriers then arrive at the electron transport chain. So these two little bubbles in blue, these are the electron carriers. And when they get to this point on the membrane, they find these great big proteins that are also enzymes that are stuck down in the membrane. So each of these proteins has a role, and they're overall what they're doing is passing electrons down the chain from one to another. And every time these electrons get passed from one stage to another, the electron is giving off a little bit of its energy. And sometimes that energy is used to pump hydrogen ions into this space. So if we think about where the energy is moving, the energy starts here, because that's where it landed up from glucose. It moves into these electrons that are then moving through the system. And some of that energy is being pushed into here. And at the end, that last electron combines with oxygen and ends up in water. So this is why you have to breathe. If you don't breathe, then there's nowhere for this electron to go. So if this electron can't go here, then that means that it's stuck in this position and you get this massive backlog, this traffic jam. Because if there's electrons here, then it can't take on a new electron from here, and then it can't take one from here, and that can't take one from here, and that can't take, and so the whole thing shuts down. It's also the same reason why cyanide is deadly, because cyanide impairs this molecule right here. And so even if you're breathing, which you are, you can't use any of that oxygen. And so you die in sort of the same molecular way, whether you are suffocating or dying of cyanide poisoning. So that's pretty morbid. Okay, so at the end of this, now all of that energy that was here is sitting in this spot right here. And the reason why that is a source of energy is because, again, this is a place of lots of charge and lots of concentration. So last week or the week before, you did the osmosis lab, right? And you found that when there is a high concentration of the same kind of thing bouncing around, there's a greater rate of movement, right? There's more movement and there's more energy when there's lots of things shoved together, because these hydrogen ions would like to be diluted and they would like to get out of the space. They're all positively charged, so they're all repelling one another. And the cell is just forcing more and more of them into this space. And so you're creating this like charged little balloon in here that just wants to burst. So that's where all of the energy is. And that's another form of potential energy. So it's not in between bonds, but it's in this concentration and electrical gradient there's this high positive charge in here, there's a low positive or a negative charge in here, and this is a huge source of energy. And this is the molecule that can use it. So this molecule is ATP synthase. It sits also in the same spot. Here's a diagram I'll let you look at on your own later that shows them all together. So this molecule is ATP synthase, and it's sitting in the mitochondrial membrane. So when you create this huge gradient, the gradient can pass back down its concentration gradient, which it desperately wants to do, but it can only do so if it moves through this molecule. So when it moves through ATP synthase has this really cool structure where at the top, it's essentially like a channel protein, just like aquaporin that lets water move through. So it's just a channel protein for hydrogen ions. And on the bottom, it's got a motor. So it's got these 4 components that act like a rotor, and it actually rotates and it clicks and turns, not clicks, but it rotates a certain amount every time a hydrogen ion goes down. And that rotation is a form of kinetic energy. So in this moment, we're taking potential energy, converting it to kinetic energy because we're moving this thing over. And that movement forces a phosphate onto ADP to create ATP. And so now there's our final source of that energy. It's back in potential energy, and it's sitting in that bond in ATP. So this is where the vast majority of the ATP is made. All of those other steps were about breaking down glucose. You got a little bit of ATP, you made all those carriers, but this is the moment where all of that energy in glucose really comes to fruition, and you get this huge amount of ATP produced. Okay, so we've been talking about glucose this whole time, but let's recognize that all of the food that you're eating can enter this system. So let me skip ahead one second. We saw something, we talked last week about sort of the way that these molecules are transformed in the body and from one source to another, and that some of them can be converted into one another, right? And all of these things can enter this pathway. So we always talk about glucose because it always enters at the top. And so it moves through the entire pathway, and it's a really handy way to see, so how does this pathway work when you put food in? But other stuff can also be put into the pathway. Sometimes they just come in at different spots. So fat, when you eat triglycerides, gets broken down to glycerol, which starts at the beginning, and fatty acids sort of come in the middle. Proteins, depending on what kind of amino acids you consumed, they're mostly not used in this because they're mostly used for making new proteins, but some of them can be used here and they can come in at different stages. So when we talk about when somebody is losing weight and they're losing fat mass, that's usually what we're talking about, right? So this fat is being processed through this system, and it's being used up for energy because the body is short on energy for other, in other forms. So your friend was doing this. They lost 5 pounds of fat because they adjusted their diet and exercise. So where did those molecules actually go? The triglyceride, the fat molecules. They were not destroyed. So where are they? No one thought it was converted into ATP, which is correct. It was not converted into ATP. The thing that gets converted into ATP is ADP. That's what gets converted into ATP. One person said they were burned and released as body heat, but that implies that the matter in the atoms just is released as heat, but it's not. So the energy of something can be released in heat. The potential energy stored in the bonds of that fat can be released as heat, but the molecules stored in that fat cannot. So those atoms have to go somewhere. They have to go into a different form. We were split between the last ones, whether it was converted to CO2 and exhaled, maybe it's back on screen, or whether they were hydrolyzed to fatty acids and glycerol. So if we look back at this, we can see that the first step of fat digestion is to split them into glycerol and fatty acids, but these are not excreted. The whole purpose of splitting this fat down into these forms is so that it can be used to generate ATP, right? So if the body is low on energy because someone is dieting or exercising a lot, then they're hydrolyzing this fat because they need things to put into this pathway. So you would not want to excrete them. So the answer was the one that is often the least logical. Those things were converted to CO2. So just like at the end of this pathway, the glucose got converted into CO2. If we look at this one, it shows us that it also got converted into water, right? That's where those atoms ended up. So when someone is losing weight, they are breathing out the fat that they burn, except now it's in the form of CO2 and water. Isn't that amazing? There have been all these studies. This is a really common question that people can't wrap their mind around. So there've been a lot of cool studies where people will go out and just like ask people in the general public what happens when you lose fat. They've even done it where they go to like college campuses and ask science majors what happens when you burn fat, and people are like, oh. So you can actually weigh yourself before bed and then weigh yourself in the morning, and you will weigh less because all night long you were breathing out CO2 and H2O, and you were in a period of fasting, and you will lose a small amount of mass because you were doing respiration. Okay, so I want to talk to you about how this all relates to metabolism and body heat and a special kind of fat. And I can't remember if you read about this or not. Did you read about brown adipose tissue? I don't think you did, so I'm going to tell you about it. So you might have heard about this because it's been sort of a hot subject of study, and we'll get to why in a minute. So brown adipose tissue is fat tissue. So our normal fat tissue that stores triglycerides for energy purposes is white. And so it's usually just called adipose tissue. But brown adipose tissue is actually brown, so that's why I call it. Brown adipose tissue is fundamentally different than other kinds of fat because its mitochondria have this special channel inside of this membrane. So I said before that the only way for these hydrogen ions to get out of this place where they're trapped is to move through this channel, and then that's going to activate this, and they're going to make ATP. In brown adipose tissue, there is a special channel that's just sitting over here somewhere, and it's going to allow some of these hydrogen ions to move back without going through this. So when that happens, the potential energy that was stored in that gradient is transformed into heat because you're moving from this concentrated space down to a less concentrated space. You're not harvesting that energy to make ATP, and instead the energy is released as heat. So this is really important for maintaining body temperature in animals that are endothermic. So you find brown adipose tissue in hibernating animals and newborn animals, mostly mammals that do this. Adults have it a little bit too. So mostly adults have a little bit like in between their shoulder blades, and there's some right here. And it makes sense because if there's anything in your body that you need to keep warm, it's sort of this part of your body, right? Other parts can get cold and fall off and you'll still be alive, but you've got to keep your core warm. So you have a little bit of this fat, and this is what it's doing. So it's not great for storage, but it's great for maintaining heat. The reason why people are interested in studying this is because there have been scientists that have shown that you can convert fat in between white and brown. So if you could give someone a drug that could convert some amount of their white adipose tissue into brown adipose tissue, they would lose weight. Because these brown adipose tissues are not meant to store fat. They're meant to like take the fat and then turn it into heat. And so this is one of the reasons why it's been getting attention in science. The other part of this is that the brown adipose tissue has this little molecule, right? Oh, wait, hold on. I want to say one thing first. So the brown adipose tissue has a special channel. There's also a drug that you should never take, and it's called DNP. And DNP is just a drug that makes a channel here. So it's doing the same function, except it's got a really, really long half-life. So this drug is available only from illicit sources. This is not a drug that you can actually get prescribed, but people do get a hold of it, and they use it for like weight training or trying to drop a certain amount of weight to get into a weight class for some event. So it's mostly used by like elite athletes who sadly die because when you take this, you impair your ability to maintain your own energy. So what's really sad is that folks will take it and think that they haven't taken enough, so they'll take more, but it has a really long half-life. And so they've taken too much and they can't now get it out of their body. And folks will show up in the emergency room saying, I feel like I'm burning alive from the inside, because their cells are doing so much thermogenesis that that's what it feels like to them. Don't ever take that. Okay, so that is one way that mitochondria can be leaky. So that's what we call them, leaky mitochondria, because they're leaking these hydrogen ions across. But some people just have leaky mitochondria. They're not brown adipose tissue. They're not taking DNP. There are genetic differences in the way our ATP is structured. So that ATP synthase molecule that's got the channel that's going to rotate, there are differences. People have genetic differences in the sequence of that DNA. And so the structure and function of the ATP synthase can be slightly different. So if you had a genetic difference that made it like not work, then you wouldn't be here. But you can have differences that make it work a slightly more or slightly less efficiently. So if you have a mitochondrion that's a little bit leaky, that means that some amount of that hydrogen ions are going to be allowed to pass through without making ATP, and they'll be released as heat instead. So thinking about this idea of how we think of metabolism, which is like how much are we eating, how much energy are we burning? If someone has a really efficient, compared to someone who has a really efficient ATP synthase enzyme, if you had a leaky ATP synthase enzyme, are you going to need to eat more or less food to make the same amount of ATP? If you're somebody that has a really leaky ATP enzyme, then that means that that's someone who has what people often refer to as a naturally high metabolism, because you're going to need to consume more food to make the same amount of ATP as somebody else. So I am blessed with a very efficient ATP synthase. So I do not have that problem. And it would be really easy for me to just eat the same amount of food as somebody with a leaky enzyme and gain weight. So there is a genetic contribution, a large genetic contribution to individuals' propensity to maintain or lose weight. And it is all about this. Well, it's not all about that. It is partially about this. So I brought you some data because this is one of the things that's directly creating your basal metabolic rate. So that is sort of where the conversations ended on the reading this week. And folks were talking about different things that might affect your BMR, your basal metabolic rate. You're going to look at this more next week. So the reflection for next week is really cool. You're going to actually be offered the opportunity to make all kinds of different calculations on yourself to see what kinds of things you can learn about your own metabolism. So this graph came from this huge study where they measured basal metabolic rate for a whole ton of people across all their lifespan. And what they found is nothing that is a surprise to anyone over the age of 20, which is that your BMR declines with age. So the older you get, the lower your metabolism is, and that is just a fact. And so it's one of the reasons why it's much more easy. You can be much more successful gaining or losing weight when you're younger than you are when you're older. So over the entire lifespan, there is a 30 to 40% decline, which is quite large. But the other thing is you can see that there's a ton of variation, right? This is a highly variable trait, highly variable. And how much does this actually matter? So the other part of the question that came up was people trying to understand like what's contributing to this and what might be contributing to other ways in which we use energy. And there was this conversation about, does it matter what you eat? Does that affect your overall basal metabolic rate? So I brought you this to pull apart the different parts of what we call your TDEE, the total energy that you're expending. So the largest part of the energy that you expend on a daily basis is your basal metabolic rate. And this is all of the enzymes that are functioning in all of your cells to do the basic things that you need to do to survive. If you laid in bed and did nothing else all day, this is your basal metabolic rate that is required to keep you alive. 70%. And this is largely affected by these things. So genetics is a huge factor. Physical condition is another important factor because the more muscle you have in your body, the higher your basal metabolic rate, because at rest, muscle cells require a lot more energy than fat cells. Past dieting also has a huge impact on your basal metabolic rate because when you lower your basal metabolic rate because of your nutritional status, so if you decrease your energy intake, if you're on some 1,200 calorie a day diet, your basal metabolic rate will plummet. After you stop dieting and you return to a higher calorie intake, your basal metabolic rate does not increase. So every time you have a massive, like a really dramatic weight loss, your basal metabolic rate lowers and it never comes back up to where it was before. It can come back up some, but it won't go back to where it was. And this is why the yo-yo dieting is so awful. Because your basal metabolic rate started up here, you thought that you needed to lose weight. So you lost some weight and you lowered your basal metabolic rate. And maybe you like gained a little bit of weight when you cut it back and you tried again. So you're gonna exercise again and you're gonna dip your basal metabolic rate lower. And it might recover a little bit, but then you're gonna diet again and you're just gonna dip it lower. And every single time you do that, it's going to be harder to lose weight and harder to keep it off because every time you do that to your body, you are lowering your basal metabolic rate. There is this really terrible, interesting study of the Biggest Loser participants that came out a few years ago, where they actually measured like how many hundreds of calories a day difference in basal metabolic rate it was. And for some of the folks that had really dramatic weight loss, like instead of having a 2000 calorie a day estimated need, now they only needed 12. And so that means that in order for that person to maintain their current weight, they can't eat more than 1200 calories a day. So this is my giant message is, dear God, don't ever calorie restrict yourself to the point where you are losing weight rapidly. So dietitians will always tell you that a healthy weight loss is never more than 2 pounds a week. And that is very high. So I always would say you recommend less than 1 pound a week weight loss so that you maintain your basal metabolic rate. It doesn't matter what you eat. So what you eat does not actually affect your basal metabolic rate other than the amount that you're eating. So whether you're eating carbs or proteins, doesn't matter. The current, like the amount of calories that you're taking in does. Where digestion comes into play is up here. So these are the other categories of things that you have a more control over. So when this is your sort of basic care, like if you're going to get up and brush your teeth and go to the bathroom and walk around your house, you're going to burn about this percentage of your daily calories. If you're eating a regular amount of food, you're going to burn about 10% of your daily calories from that. And then this little tiny sliver up here is actually, oh, I went and jogged around the block. So your exercise is a pretty small piece, but it's the piece that you are completely in control of. And so that's why it's the piece that you can modify most readily. The digestion piece is impacted by what you eat. Some foods take more energy to digest. And so the question in the reading was, should you eat a bunch of protein to increase your metabolic rate? Eating protein does take more energy to digest than eating carbohydrates. But I looked up this one study and it said that it was probably about a 15 to 30% more energy that it might take to eat protein than carbohydrate. But if you look at how tiny this piece of the pie is overall, increasing it a little bit isn't going to change your overall daily expenditure very much. And so the idea that like I should just eat a ton of protein because it's going to help me lose weight is not really a very effective thing to do.